Aloha kako. It's so nice to see everybody here. This is a very exciting event. This is bringing science to the community. As you know, Malama Maunalua has been in existence for almost 6 years now. When we started, we knew intuitively, which is why we got together, that Maunalua Bay was in trouble. But when we looked for the science on that, there was none, or almost none. So in the intervening time, we have looked at historical photos. That's why I threw a lot of these up here, a lot of historical photos, archival information, maps, both old and new. We did oral histories of people who have been around here forever. And we determined that indeed, intuitively, our intuitive feeling was absolutely true. Maunalua Bay is in a lot of trouble. And we had 3 big threats: alien algae, sediment and pollutants, and there's seats right up here. And basically overfishing. So, in the meantime, because of this community's interest, there has been quite a lot of science that's happened in Maunalua Bay. And that's what we're going to bring to you tonight. And we've put a lot of pressure on our 5 presenters to make short presentations of a lot of work. They're going to really jam it in there. And we'll have a short period of questions after each presenter, and then hopefully, if we're right on schedule, they can all come up here and we can have questions at the end. And then after, for anybody who still has the energy, we'll go over to Roy's, and which is right next door, downstairs, and we can talk to the presenters and to each other. So before we get started, I would like to thank NOAA for providing this fabulous venue, and especially Jeannie, Jeannie, I forgot her last name, but who has put this completely together for us. We have Olelo here tonight. Kamuela and Scotty have volunteered their time to come film this, so it will be on public access TV. So thank you, Olelo. And I guess I think that's good for right now. I think we'll just get right going. Does anybody have any questions before we get going? So we're going to start off with Chris Ostrander. Chris is a director of the Pacific Island Ocean Observing System, and we're going to start off with very big picture. So Chris, if you could, yeah, thank you.
So good evening, everybody. I'm really happy to see this many people here, and I hope that I can do the topic justice, and give you information that you find valuable and useful for your interests in Maunalua Bay. I'll start off by saying I hope I don't disappoint you. I'm not going to actually present any science. What we do, though, is integrate information from various scientific efforts and make it available to the broader community in a variety of different formats, in a variety of different levels of processing, to assist in informing decision-making related to management or conservation or just daily use of our resources. So my name is Chris Ostrander. I'm with the Pacific Islands Ocean Observing System. It is an interagency, multi-level partnership that's based at the University of Hawaii, but includes 18 different partners around the region, of which Malama Maunalua is one of our signatory partners. So I'm going to give you an overview of our program, and delve into some of the data systems and the data visualization and the data access we have as it applies to the region, mainly as an introduction. We don't have too much time tonight. I don't want to bore you with too many details, but hopefully I can give you enough information to whet your appetite, and I'm happy to follow up with everybody individually or as a group outside of this venue. So we are part of a national program called IUS, the Integrated Ocean Observing System. And IUS, in the most simple way, is meant to be the ocean version of the National Weather Service. So we provide ocean information to inform a variety of stakeholders in a variety of sectors. And I won't read through this slide. You can see from marine operations through climate and fisheries and ecosystems, all the way down to planning and policy decisions. Our aim is to inform all of those. We do that as a partnership of 17 federal agencies at the national level that then work with 11 different regional associations that span the entire US EEZ. The regional association out here is the Pacific Islands Ocean Observing System, or PAC-IUS. We cover not just the entire Hawaiian archipelago, but all of the US territories and possessions in the Pacific, as well as the 3 nations in free economic association with the US, Palau, the Federated States of Micronesia, and the Marshall Islands. So our mandate is to build an observing system that measures upwards of 26 core variables from the high water mark out to 220 miles, which actually amounts to about 30 million square kilometers. It's a huge area. It is a massive extent of ocean. And when we started the system, we knew there was no way we could do everything for everywhere. So we focused the majority of our efforts on the south shore of Oahu. With those efforts, we targeted 4 main areas: marine operations. This is ocean safety, efficiency in transportation, sort of forecasting and monitoring of waves and currents and ocean phenomena; ecosystem information; coastal hazards, so erosion and inundation and shoreline change; and coastal water quality. Tying all 4 of those together is numerical modeling and data management and visualization. We spend the vast majority of our effort on those 2 things, on modeling and on data integration. And that data integration is what I'm going to delve into tonight. We also work with education and outreach and governance and management to really run a full system that covers all aspects of ocean observing and oceanography in the region. To do that, we use a wide range of tools. We have cabled ocean observing systems. We have buoys. We have water level stations and tide stations. We have deep water moorings. We have land-based terrestrial systems, shallow near-shore deployed water quality sensors. We tag and track large pelagics, sharks and tuna and turtles and monk seals. We use deep water ocean gliders to measure the ocean biology, chemistry, and physics down to 3,000, 4,000, 5,000 meters. We use ships, we use coastal cameras, we use LiDAR, we use underwater submersibles, little tiny autonomous submarines, we use aircraft, and we use surface drifters. We deploy these instruments all throughout the region. The vast majority of them, though, are deployed here off of Oahu and in the Hawaiian Islands. We also run numerical models related to wind, to waves, and to ocean circulation at a very broad Pacific scale, and then at a much higher resolution along just the south shore of Oahu and for the Hawaiian Islands. And everything we do, all the data we generate internally, all the data we collect from literally thousands of outside sources, is all available from one website. So pacius.org is an entry point to a lot of different ways to get data. You can look at conditions at a glance, very quick conditions for waves and tides and currents and weather just from the landing page. You can delve into real-time observations here in Hawaii and around the Pacific. And you can look at some of our value-added refined products for freshwater plume dispersal, for trajectories in the coastal ocean of where things shall go, high water level forecasts for coastal environments, ocean circulation forecasts. We have a variety of things available as refined data products. We also, and I won't go too much into this for technical users though, we have very technical interfaces for accessing modeling data, for satellite data, for all of our deployed single point systems around the region that are measuring water quality or tides or currents or waves. We have huge data archives. We're actually serving now about 50 terabytes of data that we generate on our own, as well as pulling from other sources around the region. The real value, though, and this is what I really wanted to showcase tonight, is that by gathering all of this information in one source, you can make it available in a common platform. And so you can start to look at ocean biology and ocean physics and ocean chemistry, whether it's real-time data, animations, spatial data sets. You can look at it in one place, in one environment, and you can start to see how data is interacting and how the physics is forcing the chemistry, how the biology is responding to the physics, and how all of that influences human activity and human use in the coastal ocean. So we've spent a lot of effort developing a visualization tool where you can access all of the data we provide. Because folks understand Google Earth and Google Maps much better than they understand GIS, we've invested most of our energy in a Google Maps programming interface. So this is what happens when you go to pacayus.org/map. Along the left-hand side, and I'm sorry you can't see it here, are a variety of headings, navigation, ocean forecasting, bathymetry, coastal hazards, water quality. There's about 15 different headings. Each one of those will expand and has dozens and dozens, in some cases hundreds of data sets within it. As you select through them, you can start to overlay things on the map. And so what I'm going to show you are just examples of some of the things we have available, focused mainly here in Maunalua Bay, but also expanding across the region. This is temperature. So this is ocean surface temperature from one of our numerical models. This numerical model is highly accurate. It assimilates data from all of our sensors in the water in real time and uses that assimilated data to forecast what it thinks is the entire ocean surface temperature. It does it at about 1 kilometer resolution. And so if you put on this overlay and you can change between temperature and salinity and velocity and sea surface height, you can click anywhere on the map and it will tell you the value at that point. From that, you can go back and look at what the conditions have been at that point for the last few weeks, for the last few months. You can download the data directly from this to your desktop if you want to manipulate it and use it for something else. Or you can click anywhere else on the map and see what the conditions are at that location. This is temperature. We also have currents. So this is the velocity, and the warmer colors here are a higher velocity. So these are stronger currents. And just like you would expect a flag to blow away from the direction of the wind, these little points are pointing away from the direction of the current. So they're flowing downstream. This we have surface currents, and we have currents down to 30 different bins going through depth bins. So you can look at deep ocean velocity, you can look at mid-ocean velocity, you can look at how things are moving between different layers, and you can see what the velocities are, the current speeds are on the surface. We also have waves. These are our color-coded wave heights. These are all from today, by the way. These are all from this afternoon when I pulled these down when I was putting this together. So as you can imagine, we have strong waves off the south shore on the east side today. So we're looking at 5.5 to 6 feet of face wave height along the south shore here, getting smaller as you go further into the bay. And again, you click on any point, you can see what the data is at this moment, and you can go back and see what it's been the last 24 hours, 3 days, 1 week, or 1 month, and you can plot how those conditions have changed. We also have integrated nautical charts, so every nautical chart in the Pacific has been digitized and integrated into this viewer. You can overlay them and you can zoom them in and out to make them more or less opaque if you want to start to look at multiple spatial layers at once. So here's a little slider. If you drag it back and forth, this nautical chart will get brighter or fade out into the background. This is a compilation of a few different data sets. One of them, the deep red colors are the tsunami evacuation zones. The more faded out red colors are FEMA flood zones, AE, and then you have AH and VE and X500. And if you click on any one of these, it's going to show you what this flood insurance risk zone is, why it's important, and you can actually drill down and see where your house is, what flood zones your house is in, if you live within a tsunami evacuation zone. And if you do, I've also put in all the emergency shelters in the region. If you click on any one of those little red buildings, it's going to show you how to get to where it is. It'll give you the address, the phone number, who runs it, where it is, and what it's called. So you can start to plan out if you have to leave a tsunami evacuation zone, where you're actually going to go. And as with everything in the system, if you click on anything, it's going to tell you what that data point or that data layer is. We've integrated a lot of infrastructure. So here I've turned on the boundaries in yellow of the whale sanctuary. In orange is the wildlife refuge in Pico Lagoon. I've turned on conservation districts. Here's the one off of Diamond Head and here in Hanauma Bay. The purple and pink lines are underwater cables. The green is bottom fishing restricted areas. And we've activated every aid to navigation in the region. If you click on this aids to navigation, it'll tell you what it is, what the frequency is of the flashing light, how deep it is, what it means, and you can see what else is next to it. So you don't just have to look at nautical charts and see what's there, you can actually pull out the data that's in them and turn them on and off and relate those to other things. This is the Doppler radar from the Weather Service, but I've also, more interestingly, turned on locations of monk seals. So we're pulling data from the monk seal research program. We track and tag sharks and tuna. We're pulling tuna and turtle data from NOAA. We're looking at getting whale and cetacean data from other sources, in addition to what we collect ourselves. All these green dots are Kermit, one of the more popular monk seals that the program tags. And you can animate where he swims, you can see where he's been and when he's been there. But more interestingly, you can turn on all the benthic habitats in the region, and you can see where he likes to hang out. You can see how long he dives, where he dives, and how deep he dives at each one of these locations. You can do this not just for monk seals, but every other animal we have that we can integrate into this. And not just as single animals and single species, but the next thing we're going to turn on is that you can pull every tuna data point we have and lay them out. And so you can start to see where species distributions are and how they change over time. And I've also, just for fun, drawn on the 3-mile nautical limit. You can overlay a variety of demarcation points in here. If you can think of information you'd like to have in the ocean, we probably have it in here, and if we don't, we would like to, so tell us where we can find it. This is surface currents. We measure real-time surface currents on the surface of the ocean over about 1,400 square miles of ocean area at 1 kilometer resolution. And I apologize for that kilometer and miles mismatch there. But these are all distinct arrows, and they're pointing in the direction of the current flow. This is from 2 days ago. This is not from today. But if you click on any point, you can see the current is moving at 0.1 knots. It's moving in this direction, and you can plot what the current has been at that point. This is useful if you just want to know what the ocean surface is doing, if you're sailing, if you're swimming, and you want to know what's happening. But more practically, this is fed in real time into the Coast Guard's operational search and rescue forecast system, and they assimilate this in real time every time they have a search. So now, rather than guessing what the ocean surface is doing, you can actually see it in real time. Below that, you might think this is Google's base bathymetry map, but it's actually the SOES, the School of Ocean and Earth Science and Technology, all of their high-resolution multi-beam bathymetry. We just stole Google's color scheme, so it's seamless to you to see, but it's much more rich than you would have as a standard layer in Google Earth or in Google Maps. Zooming out a bit further, this is satellite data from MODIS, which measures chlorophyll in the surface ocean. We have every US-based global scientific satellite feeding real-time data into the system. So you can grab the path and the swath from today. This is a monthly swath, actually, so you can accumulate the composites for weeks, the composites for months. You can do it for every variable that NOAA and NASA release and measure. And you can do it on the fly. So all of this is done as you select it, and is transferred in real time, the most recent data set. And again, click anywhere and you can see what the values are. And the nice thing about the satellites is under Show Data Plot, you can click All. You can see every data point that's been collected by that satellite in its entire life history at that location. So you can start to look at changes in chlorophyll, changes in sea surface height, changes in temperature over many, many decades. This is temperature. This is from today. This is the daily sea surface temperature for the entire globe. Everywhere you don't see temperature is where there's too many clouds to measure. But you can see where we fit somewhere in this semi-warm band. But it's interesting, you can resolve the ocean waves coming off of Peru that really drive and indicate El Nino and La Nina conditions. And you can integrate this with many, many other data sets. These aren't It's not set up so you can only look at one thing at a time. You can turn on as many things as you want, and you can zoom and fade them out so you can see how they interact. A few more things just to point out. Everything shown here is a real or near real-time data feed. The majority of these points on this map are things we own and things we run. The only thing that isn't are all the little beakers. The beakers are the city and county and the state's water quality monitoring locations, but we take that data in real time as soon as they release it, so it's available through our system. And I have to give credit to USGS, which runs the stream flow and rainfall gauges. But we are working to partner with them to expand those networks under our support throughout the region. But all of these things, if you zoom in to Maunalua Bay, we have some. We admittedly don't have a lot. But what we do have is a real-time water quality station right here off of the harbor. And this is from this afternoon. Here's the variables it measures, and unfortunately it doesn't come out very clearly. But these are available on the web in real time, all day, every day. You can log in and see exactly what the water conditions are and how they're changing. And to give you an example of how things actually change, all of this down here at the bottom is turbidity. And you can see there's a fairly consistent cycle until we have big wave events. When you have big wave events, you get the resuspension of sediment and sand. And this higher level of turbidity is all from that wave event we're having right now. But you also usually see a decrease in temperature at the surface as you mix up some of that deeper, colder water. So you can watch in real time wave events. You can watch storm outfalls and plumes coming off of the harbor after big rain events. We're expanding more of these stations around the region within Maunalua Bay and outside of it. But certainly if you have suggestions or wants and desires for where you'd like to see more information, we're happy to oblige as best we can. I went a few minutes over, I apologize, but I'll stop there, and if you have any questions now, I'm happy to get into them, otherwise we can chat later.
Will I be able to go to that site and find out when I have to abandon my house due to the rise in sea level? If my grandchildren need.
I could help you with that, but that's not something we want to predict for people. The information is there. From the website, you can determine where inundation zones are. You can determine historic rates of sea level rise. You can look at what expected inundation lines are for wave and water level events. But we aren't going to get that specific, unfortunately. Ma'am?
After the tsunami in Japan, we heard that there was a lot of debris that had gotten into the ocean and it was slowly moving its way across the Pacific,
Right.
but that it would turn around and come back again and hit the eastern shores of Hawaii. Can you give us more information as to where that is and what we should expect?
Yeah, so the question is related to, just to summarize for the cameras, the question is related to the debris field from the March 11th tsunami in Japan. The ocean currents in the northern Pacific form a gyre. They form a very, very large, very slow-moving vortex, or whirlpool, for lack of a better term. So anything that gets dropped into that circulation will usually go up and around towards California and then back down, and usually scrapes along the Hawaiian Islands from the eastern side. We do get debris often along east-facing shores because they tend to be more energetic and because they tend to catch some of that drift. No one is really sure how much debris came off of Japan during the tsunami and how much of it will stay afloat and for what time. But Nikolai Maximenko, who released that debris field model, which some of you may have seen populated on CNN, is our Pacific Ocean circulation modeler. So he runs our Pacific Basin surface circulation grids, and to our best knowledge, that is legitimate. So there is debris from the tsunami. It is in the ocean. It is migrating. But no one at this point has funded a search to see how much of it there is or how long it's staying up. But certainly you will find something from Japan at some point, somewhere. Wonderful. Thank you.
Thank you, Chris. That was phenomenal. What we did ask the presenters was, when they present their work, is also to note how it pertains to Maunalua. And Chris certainly did that. And it's very fun to go on the site and look at that recorder, that's that water quality recorder, and watch that data go up and down. I was wondering, Chris, if you felt if you saw any alarming trends in any of that.
I haven't yet, no. No, I mean, you see, from that sensor, you definitely see storms, you see high wave events, but I haven't seen anything else that really stands out at this point. The real power of that sensor, though, is not just in that short-term observation, but the long-term 5 and 10-year time series.
Right.
One thing I forgot to mention is that when we put these out, we leave them there, and we plan to leave them there forever. So you'll be able to look at that time series change in temperature or water level or wave intensity over many, many years and see how things are shifting.
So this will be really important for climate change
Definitely.
monitoring, I imagine. Okay, well, thank you very much. Our next presenter is Alika Winter. And Alika is his day job, night job is a fireman. And he also is our Malama Maunalua coordinator and the Malama Maunalua Marine Resource Manager. So, Alika, thank you very much.
Aloha. Aloha. All right, so my name is, can you hear me? My name is Alika Winter. I'm the Makai Wash coordinator. Oh. Okay, my name is Alika Winter. I'm the Makai Watch coordinator and the Marine Resource Manager for Malama Maunalua. What I have here tonight for you guys is a Pico Nearshore Monitoring Report. And this was an effort by the Makai Watch volunteers of Malama Maunalua, who are the real champions of all this effort that we've been doing, because these guys show up day after day, week after week without getting paid, and these are the guys doing all the work. So real quick, could I have some of the Makai Watch guys stand up? I see Richard, Linda, Ralph. Just stand up real quick, you guys, because you guys have done an amazing effort. Oh, there's Marcy. Ray's out there. So I wanted to thank you guys personally for all the work that you've done over the year. Check. So I want to thank you all personally for all the work you guys have done for almost 2 years now. So all of these, all this effort is because of them. So what we have is a PowerPoint. We have a 54-page report if anyone needs more detail. But what I got for you tonight is a quick and dirty. So basically in the year 2010, we knew there were going to be some major changes in the Pico area. Number one, the Great Hookie, where Pono Pacific and Malama Maunalua volunteers will be removing alien algae, 26 acres to be exact, and sewer line repair. If some of you are familiar with that sewer line repair, right adjacent from Pico's, right up under Kalaniani Only Highway, there's an aged and broken sewer line, and you can see me later for the hypothesis on that. But here's the area we're talking about. Pico Beach is right there in the middle, Pico Lagoon on the right, and Kalauhaihai Pond right there under the circle. And as you can see, some of that is the alien algae removal. So we had a hypothesis. We figured that the reduction of alien algae and the return of fresh water would result in a more natural environment, which would result in more fish and native algae. So the only way you can test that is with data. And that's where we found out that data is really important. With data, you can create a baseline, you can document changes, you can measure those results, and you can connect with other efforts that you guys are doing. In our case, we're doing fisher surveys and working with the enforcement forces. So all this information works together. And for this timeline, we started our monitoring back in the spring of 2010, and we're planning to continue a 2-year minimum until the spring of 2012. Here's our timeline. The blue line below is when we started our monitoring. The green line is when we removed the alien algae. And the yellow line is when the sewer line was repaired in the mid of October of 2010. And these are some points. The yellow line along Pico Beach is where we do fish counts. The green lines are where we run benthic surveys. And the blue points are where we do water quality surveys. And you may be wondering why there's 2 blue points in what looks like land. That's where Kalauha'iha'i Spring is. And here's one yellow fish count line we do inside Pico Lagoon. So for our water quality, this is what our data looks like once we enter it. And it's not very exciting. But the reality of it is this is what Kalauha'iha'i Spring looked like before they fixed the sewer line. And here's what it looked like after they fixed the sewer line. And here's a little more colorful graph. The blue lines show the salinity within the pond, and the warmer color lines show the salinity in the nearshore environments of the pond. So what you can see is, right when the sewer line was repaired, that dashed line, immediately it dropped from a brackish environment to almost a pure water environment, freshwater environment, which is pretty staggering immediately. And it also stabilized the salinity in the nearshore environment. And before I ask this question, I just want everyone to take notice of that blue water there in the ground. That's crystal clear blue water that's been digging up. Can anyone that's not one of our volunteers or with Malama Maunalua, can anyone tell me where or when this is? This whole room. I know you guys must know, no? Okay, this is a Kalaniane'ole Highway, somewhere along the mid-1990s, I'm guessing, when they were widening the highway. What you see here is where the sewer line, the sewer line was running. What they're doing is they're putting all the utilities underground. See the utilities above ground? On Kalaniane'ole, all the utilities are below ground. So what they had to do is they had to dig a trench to sink all the utilities underground. And what happened was they found a lava tunnel that was feeding Kalauha'iha'i Pond. And this is what happened when they ruptured the lava tunnel. And these are all, they call them super suckers. These are 4-inch suckers sucking constantly to get rid of all that water. They estimated between 300,000 and 1 million gallons a day was coming out of that lava tunnel into the pond and into the nearshore environment. So what they ended up doing was sealing the lava tunnel with a 19-foot concrete sleeve. That way you can put all the electricity and the phone lines and everything underground with no problem. The next area of monitoring we did was juvenile fish monitoring. Here we have a little picture of a baby mullet. This is a ama, the Hawaiian mullet. And we use belt-style walking surveys. So in this case, you take a group of volunteers and you walk along the beach. One would serve as a fisheye, the other would be the recorder. And every time you see a group of fish, you call it out, the other person would just mark it down. And inside Pico Lagoon, on an average day, we might count 3,600 mullets and over 2,000 opai. And an average day on Pico Beach, they counted 3 mullets and 0 opai. Can anyone tell me the difference between Pico Lagoon and Pico Beach? Any difference? Nobody. Well, one major difference is Pico Lagoon, you need a permit to enter, where Pico Beach has unlimited human access. And here's an example. The solid black line shows the numbers within Pico Lagoon. They pretty much are between 2,000 and 10,000 pieces, and where Pico Beach has anywhere from 12 to 250 pieces. Kind of a staggering difference in the amount of fish. And the overall composition inside Pico Lagoon, about 3/4 of it is ama, at least on the line we ran, and about 1/4 opai. where Pico Beach was more diverse, had a hole hole, ama, nehu, opai, tilapia, and some unknown or other fish. Another thing we found out that the average size was bigger on Pico Beach, while the average range was bigger in Pico Lagoon. So in Pico Lagoon, we might see a 1/2-inch mullet, but we might see a 1-foot-long mullet, where actually Pico Beach just had bigger fish. Inside Kalauhaihai Spring, what we found is 100% invasive fish, about 1,000 tilapia and about 1,000 mosquito fish. But recently, on a side note, we released a couple of hole in there, so now we get a couple of hole in there. But don't tell anyone. Okay, the last area we did was benthic monitoring. We ran 4 transects right through the Great Hookie project. Where each water quality mark was, we ran a transect straight south. What we were looking for was natives and also invasives. Some of the natives are the Hawaiian seagrass, the halamida, the caulerpa, the padina, the spiridia, the dictyota, just to name a few. And some of the invasives on the top, gorilla ogo, what we call prickly, mudweed, and hookweed. And what's interesting is this first graph on the top was before the Great Hookie started. And on this side is toward the beach, and on that side is going out towards sea. And what you can see, that magenta colored line is mudweed. And as you can see, a majority of mudweed covered the area. And this is right toward the end of the Great Hookie project. And as you can see, a majority of sand and mud and rock dominated the area, and almost all of the mudweed was taken out. And these pie charts also back that up. This one on the far left is the first transect we ran, almost all mudweed. And the last group of pie charts in the end is toward the end, where it's mostly sand rock, and you can start to see some of the green coming back, some of the natives coming back into the area, which is good. And here's before the Great Hookie, during the Great Hookie, and after the Great Hookie. And here's one of the native colonizers, Spiridia. We're already starting to see a lot of this one come back, so that's a good thing for the area. In conclusion, alien algae was significantly reduced. Freshwater was returned, but not to its original amount. Pico is a more natural environment, and native algae is returning. And the native fish numbers haven't yet increased, so probably better management on our part might be necessary to bring the fish back. So I want to thank everyone for coming tonight, and thank you to all of Malama Maunalua's volunteers for their tireless and ongoing efforts. Mahalo. Any questions? Okay, thank you.
Thank you, Alika. The results of this volunteer monitoring program are dramatic, particularly in that the repair of the sewer line resulted in a terrific change in the freshwater infusion, as Alika said, in that pond and in the nearshore water. This will be hopefully important in the future when the Department of Transportation or other agencies develop the nearshore waters and how they, what they do with the spring water that they come up against. Okay, our next speaker is Sean McDuff, and Sean is a graduate student. He's working with Dr. Bob Richmond at the Koala Marine Lab. Sean works on, or that program works primarily on coral reef and pollution issues. So Sean, thank you very much for coming.
Good evening, everybody. My name is Sean McDuff, and I work out at the Kilwalo Marine Laboratory. I'd like to thank the organizers for inviting me to speak tonight. I usually give this talk to other groups, not members of the actual community where I'm working, so there's a little bit additional pressure put on by knowing that this is you guys' backyard. So if I mispronounce any word, just shout or throw something at me. So I'm going to be talking about the sediment dynamics in the bay and what happens to sediment after the algae has been removed. So this picture here just shows the uncleared and cleared areas during this algae removal. So there's a very distinct line. So just a bit of background. I'm sure you all know this already. But Maunalua Bay is a very urbanized watershed. There's a bunch of houses, and if we look at the maps outside, the watersheds over here, they go up even further. And along, with coastal development, you usually get some sort of impact on the coastal resources on, like in the beach. And you'd expect some decline in habitat and resource quality. And with that, here in Pico, we get this Arvenvilia amadelpha, this mudweed in dense patches associated with a lot of sediment in the area. And I like to say that this is a size 12 booty, about 5 inches deep. So there's a lot of mud in this area. So, and if we want to recover reefs, we have to deal with this issue because corals can't obviously grow in this type of environment. And Malama Maunalua is I think it's a model type of organization. And here in this area, the community, with input from its partners, decided that, the resources could be better. And instead of fighting each other and blaming each other, they all decided to come together and fix the problem, which is very, you don't see that a lot in other groups. So, good job. And they, with all their partners, secured money to remove about 22 acres or 2,000 tons. That was, I guess, their goal of algae in this area. And it took a lot of cooperation from, federal agencies. Here's Mr. Jerry Davis talking to everybody about Army Corps design, I guess. Here's Alyssa Miller and Dr. Bob Richmond and community members. So every chance they get, it's a learning opportunity, and it's just a overall good. It's a good partnership. And this is just a background on the project. This is how much tonnage was removed. This is from the Pono Pacific website. I'm sure this numbers might be a little bit different. 25 acres cleared from the lagoon. And basically what the goals was to restore the coral reef. And because the mud is gone, you can expect maybe seagrass and coral recruitment in the bay. And those are very good goals to have. And so this is how the algae was to be removed. Through TNC, as science advisors, they decided to remove 1 acre plots in this fashion, which allowed for us science guys to ask some questions and do some experiments. So some of the questions we had were, are there any differences in the amount of sediment in cleared or uncleared plots? Are there any trends over time? Is the mud flushing out after the algae's been removed? And under what conditions does flushing occur? So these are some of the questions we had going into the project. So this is how we design or plan to sample the area. We randomly chose 5 points in each plot, cleared, and the green are in the uncleared areas. And this is what we used to compare later on in our graphs and in our analysis. So we have 5 plots in each block, and we measured that. We collect data on all the 60 in one day, which takes about 5 to 6 hours. So it's a long day. And this is how we do it. We use this resuspender box developed by our colleague Eric Wolanski out at James Cook University. So we put that on the bottom, we stir up all the mud inside, and we use this turbidity probe here to measure the amount of mud in the box. And we do that 60 times throughout that 22 acres. And then we let that take about 2 minutes for the mud inside the box to settle out, and then we graph that change. So once you pump it up, the amount of sediment goes up, and over time it slowly goes down. And the sediment that falls out first is all the bigger coarse sediment. So this is more your sand, your debris, all the big stuff. And then all the fine sediment takes a while to settle out. So that's this red curve here. And again, with help from Dr. Wolanski, he developed some models to turn this into actual grams per meter squared, or actual amounts of sediment in the area. And from there, we run our statistics to calculate all our averages and all our confidence in our errors. So here's one of my few graphs that I'm going to show. Here's of course sediment in the cleared and uncleared areas, and just to note that there's a lot of coarse sediment in the area. So your sand is still there; it hasn't gone anywhere, and by the looks of our data, it's not going to go anywhere. The thing that is moving though are the fine sediments, which is the blown up version of that graph. And like I said before, in the clear areas, there's significantly less fine sediment than in the uncleared areas. So if one of the goals was to restore coral health, you have to fix the bottom, because corals can't settle on mud. So by removing the algae, the sediments is going out. So that's a good thing if you want corals to come back. And another thing we looked at was the flushing time, or how long the sediments stay in the bay or in that area there. And I'm not going to go into this a lot, but this is the equation I used to determine how long sediments stay in the bay. And with this equation, figured out that in the cleared areas, without waves, without large wave events, it would take about 6.5 years for the sediment to be exported out of the bay. And with waves, like we had in September or October, it kind of knocked off a couple years off of that. So with clearing and with waves, you can expect around a little bit over 4 years for the sediment in this area to be exported. So, it's not going to be overnight. But in the uncleared areas, when there's no waves, there's no flushing, you got a positive slope, and so there's net accumulation in this micro area. But the good news is, or not good news, with waves, you don't get accumulation anymore. You get some export, but this is a lifetime or maybe a couple lifetimes. So from this area, you would expect it to take about 90 years, roughly, for this stuff to get out on its own, without addition of new sediment, without things changing. So what does this mean? It means that this mud is going to stay in this area 20 times longer than if it were cleared. So, and here's the graph summing it all up. This is uncleared areas, and this is the fine sediment before and after waves. We have our days on the bottom, and our concentrations on this vertical axis here. And this line here is a positive trend. You can't really see it, but it's actually going up. So this is that situation where you get no flushing, it's all accumulating here. And with the waves, if we can live that long, that's how long it's going to take for that stuff to be exported. And here's kind of the good news. This is Pico Lagoon, recently cleared. We have about 25 acres. And this is what happens when there's no waves, about 6.5 years. And with some waves, down to 4 years. So we're going from this to this, which I think is a good move if we want corals to come back. Because corals take a long time to grow. So I think that's good news for all of us here. So what does this all mean? That there's significantly less fine sediment in cleared versus the uncleared areas. And that means that, you know, algae does play a part in holding down that sediment. And I didn't show any graphs on this, but from what we saw, that after you pick the algae up or remove it, that it's effectively released and it just moves around in the area. So it's not going anywhere, it's able to move, but it doesn't move out of the area. And this can be, you know, we can fix this through creating, you know, corridors for the algae to actually move. So instead of having a clear box, maybe creating some way to link the box or that clear area offshore. So that's something we can, you know, kind of discuss for future management of the area. And that algal removal and wave events accelerate the export of fine sediment in Pico area, but you know, this can be used in other areas. And accelerated by how much? Like two years in the cleared areas with waves, and about 90 years if you just clear the area. So in conclusion, the algae play a role in sediment retention. That swell, in this case soft swell, is important for, you know, export. And you know, Maunalua Bay model organization, you know, so we're on track for restoration, and you know, it's a good example of successful community-based management. And the great hooky, I actually called my friend yesterday, Hawaiian friend, and said, hey, what does hooky mean? Because I wasn't sure. So he said it was for hook, I guess, is that correct? So it makes sense. So the great hooky was a great success, and it's a necessary first step in the restoration of Maunalua Bay. And I'd just like to thank all the people that helped out, all our PIs, and Dr. Layton Taylor is in the crowd, very helpful, resident of Pico Lagoon, all the Richmond lab members, and everybody that played a part in for this research. Thank you.
Are there any questions? Any questions for Sean?
Sir.
I have one quick question. Your model to look at the detention time in the fine sediments, do you hold constant any input for sediments, or do you negate that input?
We don't look at it right now. The overall trend kind of stayed the same. So if there was input or output, you know, it kind of negated itself in this area, but that's something we might have to look at in the future. But I did think about that.
I have a question, Sean. Did you see anything dramatic when the tsunami hit here? Did you see any changes?
The tsunami would have been right around here, 212, day 212. So if you look at some of the points, not really. I know Dr. Taylor had some debris in his backyard from the tsunami, but that doesn't show.
Write it down next time.
I think from what Chris Ostrander said this earlier, maybe we can look there for tsunami impacts. But we didn't see any in our data.
Any other questions for Sean? Not? Okay, thank you very much. Next, I'd like to introduce Ken Lognecker. Ken is a research assistant at the Bishop Museum, and we're talking about the effects of mudweed removal on invertebrates in Maunalua Bay. Okay.
Okay, thanks a lot. I'm here to talk about how this habitat restoration effort affected the animals that live in Maunalua Bay. My colleagues and I at the Bishop Museum were concentrating on that area where the invasive mudweed had been removed. This mudweed first showed up in Hawaii about 30 years ago. We hadn't seen it around here before then. And since it appeared in Maunalua Bay, there's been a drastic change to the shallow water environment. And we know from old records, historical records, that there used to be plenty of seagrass out there, there used to be plenty of native seaweeds, sandy habitats, and rocky reef. And of course, more recently, the community groups have been concerned that mudweed has basically taken over the area. It dominates that habitat, or dominated the habitat. And there may have been an associated decline in the number and types of native species that you would find out there. So again, as we've heard earlier today, Malama Maunalua and the Nature Conservancy removed mudweed from over 22 acres of the bay in the hopes of restoring that to a more natural state. So what we were doing was looking at the invertebrates to see whether the sorts of desired changes have happened. And I'll introduce you to the invertebrates that we were considering in just a moment, but first I'd like to talk about exactly what it was that we were asking. Okay. We had two purposes. The first was just to evaluate whether this mudweed removal was an effective habitat restoration technique. Now the near-term goals, if you read the scientific literature, the near-term goals of habitat restoration are to provide some sort of a suitable living space for the species that would normally occur in an area. And this is done by attempting to restore or to return an area towards its more natural state. And you evaluate whether that's been done by comparing the restored area with some nearby area that's still considered to be in its natural state. So that's number one. The other thing we wanted to do is, because we were looking at these invertebrates, and because they're important fish food, we wanted to see if we could say anything about how this mudweed removal may have affected the fishes in Maunalua Bay. Well, I'm going to go back and talk a little bit more about this habitat restoration idea. And first I need to talk about how we're going to judge whether habitat restoration has been successful or not. And what we're going to be looking for is, and I'm going to put this in quotes again, an acceptably small difference between those places where mudweed has been removed and some of those nearby natural or native habitats. And by acceptably small, we're going to be looking for three criteria to be fulfilled. So please keep that in mind as I go on through here today. Okay. What types of invertebrates did we get then? I'd like to introduce you to them briefly. We found mostly ones belonging to one of three major groups. We've got a lot of mollusks, or clams and snails. And again, just to give you an idea of their size, please look at these sand grains in the background, and you'll get an idea about the size of the animals that we're talking about. We got a lot of polychaetes, or marine worms. And again, for scale, you can look at the pores in these sponges right here, just to give you an idea about how big they are. Our other major group are called parachaetic crustaceans. I don't have a common name for these guys, but they're relatives of crabs and shrimp. Okay? But again, these are small guys. So here are individual plant cells, and here are individual sand grains. There's a little head of the animal sticking out right there to give you an idea of what size invertebrates we're talking about. Okay, now that you've met who we were looking at, how do we figure out whether this habitat restoration was effective or not? The first thing we did was a statistical technique called ordination. And all this does is some fancy statistics. It looks at all the types of invertebrates we got and how many of each type we got. And then it gives us some information that we can plot out on a graph. And what I'm going to try to do right now is show you what we would think of as our best case scenario. What would be the best possible support that we had effective habitat restoration out there. Then what we'll do is come back and compare our actual results to those best case scenarios. So the first thing we might expect is, because mudweed is not native to Hawaii, that it would be on the opposite end of the spectrum from all those native habitats that we were using for comparison. So here we just place them on opposite ends of this graph. The next thing that we would be hoping to see then is that removing mudweed caused some sort of change that made those removal areas more similar to one of the native habitats. And what we might even expect to see is some sort of chronological progression from 3 to 6 to 9 months, suggesting that mudweed removal was actually pushing these cleared areas towards some sort of a hypothetical native habitat. That's what we're hoping to see. Now the danger of this sort of an analysis is that we're probably not very likely to see exactly what we were hoping to see. Okay? Anybody that's ever dealt with animals and biology, and especially ecology, the patterns always get a lot more complicated than these simplistic ones that I'm showing you here. The danger is that when it doesn't look exactly what we would like it to look like, maybe we would interpret it to be like, it's pretty close to what we would like it to look like. And then the danger is that you see what you want to see. So we tried to remove some of this subjectivity by using another technique called cluster analysis. Here I have those mudweed and hypothetical native habitats again. And this time you can see that they're all joined by a line from one to the other. And an easy way to interpret these cluster analyses is to pretend that you're looking at one of those mazes that restaurants sometimes put on the back of a keiki menu. You know, where you have to get from point A to point B without picking up your pencil. Okay. So what we're going to do now is to find out how similar or different habitats are from one another. We're going to trace a line from one to the other. The lower on this graph you have to go, the more similar they are to one another. So these native habitats B and C are very similar to one another. The higher you have to go on this graph, the less similar they are to one another. Okay, so mudweed in this case, in this hypothetical example, isn't very similar at all to our native habitats. Okay. Now what might happen when we throw those cleared areas into the mix? What we're hoping to see, sorry if that's a little bit lower than some of you can see. What we're hoping to see, well here's the cleared areas that I threw in here, is that they would be very different from the mudweed. So again, to get from any one of these to the mudweed, you have to go all the way up here to the top, okay, indicating that they're not very similar at all. On the other hand, you just have to cross this kind of low bar to get to one of the native habitats. Okay, so that would show us or suggest to us that these cleared areas are in fact very similar to one of the native habitats. Okay, that's what we would like to see. What did we actually see? Actually, it wasn't that bad. Here we go. I'm just going to show you the mudweed and the native habitats again. Here's mudweed kind of more towards the middle than where I suggested it might be. Okay. Up here are the two of the native habitats. Sand is actually pretty far away. Grass, maybe you consider that far away. I don't know. Again, that subjectivity starts coming into play here. Surprisingly, over here was the limu, kind of really close to the place that we were expecting the mudweed might show up. But what happened when we threw the mudweed and the cleared areas into the mix then? It turns out that they all occurred way over here, okay, really on this extreme edge of the graph. That tells us, I think, two things. One is that they were quite different from the mudweed, so we've satisfied the first criteria that we were looking for successful habitat restoration. The other is that they're pretty close to one or maybe two of these native habitats, satisfying the second criterion that we were looking for successful habitat restoration. Again, the danger here is that I'm going to be seeing what it is that I like to see, to tell you all that you did a great job. So let's see if the cluster analysis sheds any light here. Again, here are all of our areas that have been cleared of mudweed. Okay. I'm going to trace a path now to the mudweed habitat itself, and in all cases you have to go all the way up here to the top. Okay. So the cleared areas are very different from the mudweed habitat. Good job. The other is that these cleared areas, the next place you have to go to get to a native habitat is over here near the sand. So just like I was suggesting on that last slide, it looks like these cleared areas are kind of close to the sandy habitat. Okay. Again, it looks like the first two criteria for successful habitat restoration have been satisfied here. A couple of things that I would like you to note, and again I mentioned it in this last letter, alluded to it, is that the native limu and the mudweed habitats actually appear to be a little bit more similar to each other than what we would have expected. The other is that these areas that have been cleared of mudweed are more similar to each other than they are to any of those other native habitats. This also suggests that maybe we've ended up with something that's a little bit different from one of the native habitats that we used for comparison. And I'll come back to both of those points in just a little while. Just going to give you a quick count of the number of introduced species we saw out there. In mudweed we saw 5, whereas in the cleared areas we saw 2 or 3. So for that third criterion, again, I think we can say that we've satisfied it. What can we say now in terms of mudweed removal as a habitat restoration technique? In my mind, based on those criteria that I showed you, it was pretty successful. It did a good job, at least for the duration of the study that we did. One thing that I have to point out, though, and I was alluding to this as well, is that even after 9 months, we didn't end up with an invertebrate community that looked exactly like one of those native habitats, suggesting that maybe we've ended up with something that's a little bit different. Alika showed us that basically a lot of the sediments were cleared out of that area and left a lot of a rocky substrate out there. Okay, and again, remember, I can't push this stuff into the rocks. We'd get out there and try to take a sample. We had to look around to the closest place that had some sediment that we could take a core in. And it just may be that these invertebrate communities were so influenced by all that rocky substrate out there that really the sort of native habitat that we might have ended up with might be more similar to one of these, maybe just a rocky reef flat habitat. But I'm speculating there, so please don't take that home as a we've definitely got a rocky reef flat invertebrate community out there. It takes a different sort of sample. Okay, I'd like to shift gears really quickly and talk about the fish now. Again, the idea being that these invertebrates are fish food. If you change the food, you might change things further up through the food chain. A couple of things I have to say. Probably a lot of people are excited when I said we're going to look at the fish, and they're thinking about these sorts of things. I'm not going to be able to say much about the big guys because I simply don't have very good dietary information about these guys. So I'm sorry if you're disappointed about that. More what I'll be able to talk about are these smaller and less obvious sorts of fishes that are all over the reef out there. These are the ones that are eating the sorts of invertebrates that we were studying, and these are the ones that would likely be directly affected by any changes to the invertebrates. The other thing that I need to confess right now is that we didn't sample fish during this study. So anything that I'm about to say to you pretty much amounts to speculation, so please keep that in mind as well. Well, what did we find? A couple of important trends, I think, with the invertebrates that might bear on the sorts of fish that we find out there. The first thing we did was to count all of the invertebrates that we found in each one of these cores. Okay. We then calculated the average number of invertebrates per core in each of the habitats. So here you can see that mudweed had, on average, the most invertebrates, all invertebrates that we identified. And you can see that it looks like in the cleared areas there were fewer invertebrates. But we have to use statistics to figure out whether these are differences that are large enough that they should worry us. Okay? And I'm going to walk you through the results of those statistics right now. And there are three important results from this graph that I'd like to point out. The first is that statistically, we couldn't tell the difference between the mudweed and lemur habitats in terms of their numbers of invertebrates. The second thing, that is, they effectively have the same number of invertebrates if you're just thinking in a statistical mind frame. The second is that for all of these areas that have been cleared of mudweed, we could not distinguish them from any of these native habitats, including that native lemur. The third thing that we should say is that the big difference that we saw, and the ones that would concern us, is that there was a significant drop in the number of invertebrates from this mudweed habitat when we started looking at those cleared areas. There were just fewer invertebrates there, and that was statistically significant. Okay? But the upshot here is that basically we couldn't tell these two habitats apart in terms of their numbers of invertebrates, and we couldn't tell the cleared areas apart from any one of the native habitats. Talking about those small crustaceans, the paracaras now, these are important food for those fishes that I showed you just a little bit earlier. We saw the exact same pattern. It couldn't tell the difference between mudweed and native lemur in terms of the numbers of paracaras in those cores. We couldn't tell the difference between those cleared areas and any one of the native habitats. But again, we saw a significant decrease from the mudweed areas to those areas where mudweed was removed. So fewer of those types of crustaceans. Last group that I want to show you are larger crustaceans. These are likely to show up in the bonefish and jack diets. Okay? But again, remember that I'm talking about things that really aren't much longer than my pinky is wide. The other thing is that I don't feel that we sampled these particularly well, so take this for what it's worth. One of the differences, in fact, the only real difference that we saw was that there were more of these larger crustaceans in the mudweed than there was in the cleared areas after just 3 months. In fact, we didn't find any of these larger crustaceans in the cleared areas after just 3 months. But what happened was, over time, 6 months we had some more, 9 months we had even more of these larger crustaceans. So it looks like the numbers are building through time. And in fact, after 6 or 9 months, we couldn't tell the difference between these cleared areas and any of the native habitats. In fact, we couldn't tell the difference between these cleared areas and the mudweed. So what does all this mean for the fishes? Again, I'm going to highlight that I am speculating here, so I have to put that word conclusions in quotation marks. So please don't forget that. What have we seen throughout this talk? A couple of things. One is that mudweed and the native limu tend to have the same kinds and numbers of invertebrates. The other thing that we've seen is that those cleared areas tend to have different kinds of invertebrates and maybe fewer of them. And what this might suggest is that in Maunalua Bay, where this habitat restoration is taking place, we might audibly expect to see fewer of the sorts of fishes that live and feed in seaweed. On the other hand, and I think this is encouraging, in general we saw that there were the same numbers of invertebrates as we saw in any of those native habitats. And what this suggests to me is that we might audibly end up with populations of fishes that are pretty much the same size as we would expect to see in any one of Hawaii's native habitats. Okay? In other words, it may just be that this habitat restoration effort worked for some of these fishes as well too. I'll close with thanks to these organizations and people who made this work possible, especially a lot of folks put photos on the internet these days, and I use a lot of them quite liberally, so thanks for that. Thanks for your attention, and am I allowed to take any questions or not?
I don't take questions.
Okay, all right.
Absolutely.
Questions if you have any.
I know when I've been working at removing alien mudweed. A lot of times I can't go through and pick out all the little critters in it. Do you think by clearing that stuff out, we cleared out a lot of the critters as well?
You might have cleared them out, but you have to keep in mind that you've taken their home away. I mean, they're in that mudweed for a reason, I think, and they like to live there for whatever reason. So you've kind of shifted the habitat, and chances are that you're going to shift the sorts of invertebrates that can live in that habitat once you've changed it. And again, it's one of the things that really was supposed to have been accomplished. You were trying to change that reef flat to more of a native community, and it seems to me that based on these results, that's what happened. I don't know if you'd shaken those invertebrates out and left them on a sandy or rocky reef flat, that would be the place that they would ultimately end up anyway. They might die, they might move away, they might get eaten by fish right away. There's any number of things that could happen when you take away their home.
When you take those cores, I would assume that when you core an uncleared area, you're getting more mud because the algae holds on to the mud. And when you go to the cleared areas, the core sample is smaller in terms of volume, so you would naturally find less animal mud in the smaller core.
Yes, but was that true? Did you always find fewer? Oh, in terms of average numbers of all invertebrates. However, there were some cases that some types of invertebrates responded very well to that cleared habitat, and I didn't show you any because I was trying to talk big picture right here. But there were some invertebrates that just moved in there and thought that was a spectacular place to live. It's just that, again, you've changed the habitat somewhat. And you're right, there was mud, but a lot of times these invertebrates are kind of living up on top of the mud and even perhaps in the mudweed itself, not necessarily down in the sediments. Right? We pushed this thing about 4 inches down into the sediments just to get a standardized sample if we could. And you're right, a lot of times we did get very little sediment in some of those cleared areas. But again, it's on an organism by organism basis, and I think in some cases we saw that there was a spectacular response to these areas having been cleared.
On your charts, 9 months was your latest endpoint or data point. Is this expected to continue through 12 and 15?
Well, someone from TNC just spoke to me and said that maybe they'd like to go out and try to hit this up again and see what's happened. We can go back to those areas that have been cleared 3, 6, and 9 months at that time, hit them again, and we would have data points that represent somewhere further in time. it's one of the unfortunate things that all this stuff always seems to come down to what funding is available, so.
You noticed that when you introduced mudweed, are the invertebrates there a different species than what you might find in the invertebrates in the more native seafood?
They were somewhat different, and again, if I can go back, if you all can bear with me till I go back to some of those ordination plots. This one, these guys are kind of grouping together here, and these guys are kind of grouping together here, but they're not actually overlaying one another. Okay, and here this is suggesting that they're about 88% similarity there. Okay. So they're kind of the same things. If you look at the top 10 species, many of those were shared between the two habitats. If you look at just the species that make up 50% of all invertebrates that we found in the cores, there were a number of ways that we could look at it. We did see an awful lot of similarity between the native limu and the mudweed. Did that answer your question? Yes.
Related to an earlier question, what did you do if you could only get an inch of sediment at a place? Not use that location?
We had to use it. So our bigger challenge was actually finding a place where there was an inch of sediment in some places, honestly. I mean, it was a very good question that you had back there. And we can only push that thing down as far as it would go down. Now, I don't want to give you the impression that we always that we never bottomed out in mudweed habitat either. Sometimes there were just large rocks down there that this thing wasn't going to fit around. There were a number of things that could have stopped us from going 4 inches into the sediment. We stopped how far we went at 4 inches into the sediment.
So the number of critters per unit volume is kind of deceptive if you're only collecting a little bit from some places and a lot in others. Or does it not matter because they're almost all in the top centimeter anyway?
They're all towards the top. And again, we were pushing in to try to get everything. After about an inch, I feel pretty comfortable that we've gotten the majority of the invertebrates. Basically, the way that I could have presented this and would have presented this is, if I did it differently, is not just number of invertebrates per core, but density, so the number of invertebrates per unit area. Okay, I typically, when doing this sort of analysis, wouldn't have considered the number of invertebrates per unit volume. So the bigger way to standardize it, the more common way to standardize something like this is how much, or what sort of densities are you getting, so considering only this area.
Can you tell me, if speculating, but I mean, does your data say anything to you about cleared versus non-cleared for larger fish? I realize that isn't what you really studied, but...
Yeah, mine don't. The best that I could get for you right there was when I tried to look at those, because I was told that there might be some interest in this group about the larger fish. And certainly I like the fish, and I'm interested in those larger guys too. So I did go back. I don't have, in fact, I was told that bonefish might be of particular interest out there. I didn't have access to any of the dietary studies that have been done in Hawaii on bonefish. So I had to go back and dig through literature from elsewhere. And what I was finding was a lot of bonefish diet studies from the Florida Keys.
From where?
The Florida Keys, off of Florida. There's a group down there that have been working down there for a long time. They were finding some fish that we simply don't have in this area. So right away we know, start questioning whether this is a good comparison or not. Toadfish were one of the most common predators for bonefish down in the Florida Keys when they started getting above a certain size. So right now we're starting to throw a little bit of a shadow of a doubt on what I'm going to be able to tell you. Anyway, they did see some of these larger crustaceans, those alphaid shrimp. Again, I was only getting little ones in these cores. But the reason that I included those was because, in my mind, those were the most applicable to what might be happening to some of the larger fish. And again, it looked like after 6 or 9 months, we couldn't tell that habitat, those cleared areas apart from any of the native habitats. And although the numbers were different at that point in time, we couldn't tell those cleared areas apart from even the numbers of those sorts of invertebrates that were occurring in mudweed.
Thanks, Ken, for that fascinating presentation, especially from the perspective of small critters. One thing, I just had a quick comment about timescale. You did mention that it took 30 years for this mudweed to dominate this place. So in terms of a response, kind of a 6 to 9 month period is very short. So perhaps it'll take a lot longer time, like Sean McDuff's presentation, probably in the order of years before we see full restoration from that. So just wanted to put that out there, that it's not going to happen overnight, and it'll certainly take a lot longer.
I would think so too. You know, if I'm right about my speculation about what sort of habitat we were looking at, and I said that maybe we were looking at more of a shallow reef flat community. The corals aren't out there yet. It takes some time for those things to reproduce, to settle out, to grow up to the size that you would start seeing more of a coral reef environment. So you're right, it is going to take quite some time, and perhaps we jumped the gun a little bit in trying to say that 9 months was going to give us that good comparison. Looks like I better get out of here. Thank you.
So this is a really important step in Malama Maunalua's effort to have a healthy day. We were told at the start that if you don't have the support of the fishermen, you're not going to be able to deal with any fishing issues. And so we feel that we're very encouraged by the concern and on the part of the fishing community and their willingness to get involved in solutions. So, so we anticipate that we'll be moving forward. I want to say that Alika Winter led up this effort. He had everything to do with developing the questionnaire and putting together the people who asked the questions, who did the interviews. And so you can ask him questions. Since Jack isn't here, ask Alika. He'll have good answers for you. We have a handout out there which has an abstract of all of the speakers. If you are interested, please pick one up on your way out. And also, I'm wondering if we could just have a show of hands of people who might be interested in going over to Roy's after. I know it's late, but I'd like to just have a little show of hands because we need to go set up some tables over there. Okay? Okay, up they go, up they go. Okay, great, thank you. We'll have a good idea of, is that good, Jennifer?
That's good, thank you.
Okay, Jennifer's gonna go stake our claim over there. Okay, we have one last presentation. This is Dwayne Mitten, and Dwayne is the science advisor for the Nature Conservancy. I must say that I don't know how to express our thanks to the Nature Conservancy. They have been an astounding partner from the very start. They put us together with other marine groups and helped us to develop a conservation action plan at the very start. That has kept us focused, and it has allowed us to be very clear about where we're going and what we're doing. It also has helped us with so much science advice, and nobody's done that more than Dwayne. So I take this opportunity to extend a special thanks to you. Okay, Dwayne.
Thank you for that wonderful introduction, Carol. TNC, I know, has been working in Maunalua Bay for quite some time, and it's a great place to do work, and the entire community are a great group of partners. It's been a real pleasure working with everybody here. I'm going to talk about fish across Maunalua Bay. I'm not going to talk about just the alien algae removal project or anything along those lines. I'm going to take a much bigger look at the stuff. It's really nice following Jack's talk, because Jack talked about what fishermen perceive and what they are seeing with their eyes out in the bay. And as a scientist, I know that a lot of times what we see isn't what's really going on out there at times. But I'm going to show a lot of data which unfortunately is going to support what the fishermen are perceiving is actually going on in the bay. So kind of in a way, that I may be ending us a little bit on a downer note, but hopefully it'll be a downer note with some hope for the future coming along with it. TNC's been monitoring a variety of sort of marine stuff for several years. We've had a marine monitoring program that works in a number of different communities. We monitor things like fish, the benthic environment, and our goal is to assess changes in that environment and in those fish stocks and so forth over time. One of the places we've been working since at least about 2007 and perhaps earlier, it actually predates me at TNC, is we've been monitoring fish populations in Maunalua Bay and actually across all of Maunalua Bay. And so today I'm going to present some of that data. Now, our monitoring program has about 5 main questions that we're interested in looking at. And I sort of focus these questions specifically down in Maunalua Bay, because that's what I'll be talking about today. But, you know, what kind of fish are in Maunalua Bay? That's a very basic question, of course. How many of those fish and how big are they? How does Maunalua Bay, that is the fish in Maunalua Bay, compare to other areas in Hawaii? How do these fish populations change over time? And then, how do management actions actually affect these fish populations? Now, we don't have enough data at this point to talk really about those last 2 questions, so I'm not going to address those today. What I'm going to focus on are really these first 3 questions. Now, for our work, we need to go out and actually collect some data in the field. And actually, I feel a little ashamed after seeing Chris's talk too, and actually Kenny's talk, because we actually even use lower tech equipment, I think, than Kenny. We actually get in the water and swim around, look at fish, and count fish, which is kind of fun to do. But we do it in such a way that we can draw conclusions about what Maunalua Bay looks like on average across all of Maunalua Bay. And we do that by selecting our monitoring sites in a very specific way. We go out and we randomly choose monitoring sites within Maunalua Bay, but we choose them such that they have certain parameters about them. They have to be between 10 and 50 feet deep, and they have to be on hard bottom. So if fish populations don't reside within sort of this particular area, we don't count them. So for example, if people want to know something about bonefish, OEO, those tend to occur inside that surf zone, up on the reef flat, shallower water. Unfortunately, we don't have the information to say a lot about bonefish specifically. Where we are sampling, however, is one of the areas where Jack pointed out in his talk, fishermen seem to have moved into recently. So it's these slightly offshore areas, that stuff over the breakers. And so our data is going to be looking, it's sort of directly related or comparable with what Jack brought up as in the areas where people are fishing currently. Now, just to give you an idea of kind of where we're sampling, we focus on 3 main areas in Maunalua Bay, and we randomly scatter points within those bays. Each one of the dots on here is a place where we went and sampled, went and counted fish in 2010. In 2009 we did the same thing, in 2007 we did it, in 2008 we did it. For this talk I'm going to focus on our 2009 and 2010 data. There's very specific reasons for doing that in terms of being able to compare across the state, but I'd be happy to talk about some of the other data afterwards. Now, something about this data that's important to keep in mind is that we've designed this sort of approach to look at the average across Maunalua Bay. We've had people come up to us and say, well, you have such low fish numbers in Maunalua Bay because you don't go where the fish are and count where all the fish are. I know where the fish are. If you were to go there, you'd see a whole bunch of fish in Maunalua Bay. But that's not our question. Our question is, how is the bay doing? So we've set up our study to look at sort of the average condition of Maunalua Bay. And by doing that, we can actually compare the average condition of Maunalua Bay to the average conditions in several other places around the state. And we actually have about 40 to 50 sites where we have data that we can compare Maunalua Bay to those sites. So once we actually have our site, how do we get our data? We put scuba divers in the water. They lay out a 25-meter line. They wind up identifying and counting every single fish that swims in sort of a box on the bottom. That's about 25 meters by 5 meters wide. For every fish that they see and count in there, they also estimate the size of that fish. Our guys train up so when they're actually in the water, they can accurately look at a fish and go, that's a 25-centimeter fish. I'm not a fish person, so I can't do that, but Russell back here is really good at it. He's one of our people. So how many fish are actually in Maunalua Bay? I can't give you an exact number or anything, but we can look at averages. There's different ways to look at abundance of fish. You can go out and just count the number of fish. There's 50 fish in Maunalua Bay. There's 300 fish in Maunalua Bay. Or you can calculate out what's called biomass. Biomass is nice fancy science talk for weight. You can actually take the size of a fish, and from the size, you can use some equations that have been worked out and estimate the weight of that fish. The nice thing about using weight is that weight takes into account both the number of fish and the size of the fish. And as everybody knows, the size of the fish is just as important as how many fish are actually there. And so biomass is what I'll be talking about for the rest of this talk. There's actually two different types of biomass that I'll be looking at. One is called total biomass. That's just how much fish is there, all fish, all species, everything we counted. And the other one is how many resource or how much of the resource fish are actually there. And resource fish are the fish that people tend to be the most interested in. So these will be the things that people want to catch to eat, the things that they might want to catch to sell in the aquarium trade, for example, although that's not very active here. But it's those fish that tend to be more important to the community than some of the other fish. It's things like goatfish and surgeonfish and ooh, things along those lines. I'm going to show a whole variety of graphs. I know a lot of people kind of shied away from them, probably because I told them to in their talks, but I'm going to show a variety of figures. And actually, a lot of them are going to look like this one. So this is scary to look at at first, but it's actually fairly easy to read once you kind of know what you're looking for. Along this axis here, this is the average biomass of fish. This is the average weight of fish that you find. It's in grams per meter squared, so that's just the weight of fish per unit area. Each bar represents the average biomass at this particular location. And there are 17 different locations spread across the state on the bottom. We have places in Maui, we have places on Oahu, we have places on the Big Islands, which is down here. And this just allows us to directly compare what we see in Maunalua and what we see elsewhere in the state. Now, here's Maunalua. I highlighted it on the figure. The average biomass of all fish in Maunalua Bay is at what I would call the wrong end of the figure here. It's extremely low, especially relative to all of these other places in the state. Now, as I said, I just selected 17 of them. When you compare them to the 40 or 50 that we have, Maunalua sits down here. It's pretty much dead last in the entire state of all the areas that we've surveyed. You'll notice that there's one tucked right here in the middle. That's actually called Southeast Oahu. That was a piece of data collected by Dr. Alan Friedlander. I think it was maybe 5 or 10 years ago. And that's effectively Maunalua Bay. So these 3 points down here are actually all Maunalua Bay. It doesn't paint a pretty picture at this point. Just to kind of throw this up, because Jack talked about this a little bit, and I was going to throw it up more for food for thought, but because Jack talked about it a lot, I'm actually glad he did. It makes it a little easier. But the areas in red over here are all areas in the state that have what I call additional fisheries management at them. These are areas that have enacted anything from a total closure to maybe a gear restriction to a species restriction. So there's been some additional management implemented at those sites above sort of the standard fishery, state fishery regulations. And what you notice is overwhelmingly, these are all the sites where basically the highest biomass of fish are occurring. as soon as you sort of cut off some of that additional management, we begin to see steady declines. So those things are on this side, sort of the wrong side of the figure. Now, I mentioned when I was talking about the methods that I wanted to talk, or that we were trying to measure average fish abundance across the state. I know TNC has heard several fishermen tell us that we're just not going to the right place to see fish. And so I wanted to sort of put this figure up and address that sort of idea a little bit more. What these are two frequency histograms, 2009 here in the blue, 2010 in the red. Along the bottom is fish biomass. And each one of these sort of boxes in here represent a range of fish biomass. So from 0 to 1,000 is this first box, from 1,000 to 2,000 is this second area, 2,000 to 3,000 and so forth. What each line represents are the number of survey sites where we went and counted fish that actually fall within this box. So for example, almost 55% of every site we surveyed in Maunalua Bay had less than 1,000 grams per site worth of fish. And so you can see most of our sites fall down in this very low area. That's that area here. In 2009 we actually had no sites that fell out here. Now in 2010 we actually had a couple of sites that had quite a bit of fish down here. These are those kind of hot spots that your fishermen will talk about. These are those areas where you might be able to go and actually find quite a few fish. They're out there. We find them just on random in our sampling. But what's important to note is that even with those hot spots there, these bars are the average biomass of fish in those given years. Even with those hot spots out there, the average biomass in the bay doesn't really change. So there are a couple of hot spots out there, but they really are not, there are not many of them, and they aren't affecting the average biomass within the bay. Now we can compare this frequency distribution to other sites, and I've just pulled Pupukea, which is one of the other sites we look at here on Oahu. And what I want you to notice is that Pupukea has almost no low spots. In 2009, we actually found none that fell into this 0 to 1,000 range. And we only found about less than 30% of our sites in 2010 were down in this low area. Now what you see is this frequency histogram has kind of flattened out. So you're more likely, when you're out there, to find spots that have much more biomass in them. And that actually shows up in these averages. When those average bars drop down, you can see that they've been shifted over. And the averages at Pupukea are anywhere from 2 to 3, maybe 4 times higher than Maunalua Bay. And just to really kind of put this in perspective, we also have a site on Kona on the Big Island. And again, you can see that there are no low areas. This has flattened out and extends quite a ways out. We have areas here that are just way off the charts, and that shows up again in the average biomass totals for the sites. So Maunalua Bay is looking very different from many of these other sites around the state. And that's consistent with some of the perceptions that Jack Gettinger was finding with the fishermen. Now, I want to pop back now and take a look at the resource fish biomass. Just again, resource fish are these kind of fishery target species, your surgeonfish, your redfish, your goatfish, jacks, things along those lines. This is the same type of figure that we saw previously with the total biomass. Again, here's Maunalua Bay. It's sitting way down on kind of the wrong end of this figure. Again, if we look at sort of fisheries, additional fisheries management, we again see the same pattern that we saw previously. A lot of your fishery management areas, again, are harboring this higher biomass of these resource species. And then as you get into the non-management, you tend to have much lower levels. So has Maunalua Bay always had low levels? Jack actually addressed this quite nicely through the fishermen surveys. This is a picture from about 1930, and I hope I've got that date right. Alika sent me the picture. But this is a fisheries catch, a one-day catch from some guys in Maunalua Bay. You can see there's some parrotfish in here, and there's some wrasse in here, and they've collected a whole variety of stuff. That's quite a catch. I don't think many people would be able to pull a catch like that out in most areas in Maunalua Bay. Now, if they're pulling these types of catches out with any sort of consistency, that's a good indication that the populations of the actual fish in the water were much higher than they are today. Just one other little line of evidence, also about higher stocks back. This is the sort of typed up rules and regulations of the PICO fishing hui, which got started in the 1950s, as I understand it. Some people in here might actually remember it, I don't know. They actually set a limit, which they thought was a sustainable limit for fishing in the PICO area, and that's put right here. And so here's what they kind of thought was a reasonable limit on fishing in the PICO area. And this is per person per day. So it's 30 fish, 5 lobsters, 5 squid, and no limit on crabs. I think you'd be hard pressed to get 1 lobster out of PICO today. So it'd be a real challenge.
Yeah, I just want to say that this came from me, and I got this out of my father's files, and this was far before the 50s, I'm sure.
Oh, far before the 50s, okay. Thank you, thank you. I wasn't sure exactly when it came from. I've been told the 1950s.
I'm pretty sure.
All right, great. All right, so what about the big ones? We see that there don't seem to be that many fish, relatively speaking, left or available in Maunalua Bay, especially compared to other areas in the state, and it looks like historically. But are they really smaller, like the fishermen were sort of perceiving out there? Well, there's a couple of ways we can actually look at fish size. There's average fish size. To get the average fish size, you just take the size of every single fish you see, and you just take the average of it. And so that's going to be kind of what you would expect on just average if you're pulling the fish out. But then there's something that we like to call prime spawners. And these have been defined as individual fish that are at least 7% the maximum size of that fish. These are the proverbial big ones that people like to catch. So if you have say a 20-inch fish is its maximum size that species can get, a prime spawner would be anything over 14 inches. So that's 70% that maximum size. Now, not only do fishermen like to get these guys, but these are also very important ecologically for population stability and recovery. And one of the reasons they're important ecologically is that larger fish disproportionately produce more eggs than smaller fish. So for example, one 27-inch omilu produces as many eggs as 86 14-inch omilu. So it takes 86 14-inch fish to equal the egg output of one 27-inch fish. And there's also some indication that, at least in some species of fish, the eggs of larger fish are better quality eggs, that they're more viable too. So not only is this guy producing a lot more eggs, but she may be potentially producing better quality eggs also. Okay, so what do the prime spawners in Maunalua Bay look like? Starting to sound a little bit like a broken record, I guess, but here's Maunalua Bay, again sitting down here. They aren't the lowest this time, which I guess is good news for Maunalua Bay. Kaneohe Bay actually had 0 prime spawners when the surveys of Kaneohe Bay were done. So there's actually no big fish pretty much left in Kaneohe Bay as far as we can tell. But again, these are very low numbers, sitting way down at the end. When we look at, again, fishery management put in here, again, almost all the fishery management sites are falling out on this side. So in these areas that have enhanced management, the big fish are actually occurring there. And that's a good thing. One sticks out right here, and I just want to talk about that one just really quickly. That's Coconut Island, which happens to sit right in the middle of Kaneohe Bay. The fact that Coconut Island has any prime spawners when Kaneohe Bay in general seems to have none, I think is fairly significant. So even though it's sitting away from its cousins over here, it's actually doing what it should be or supposed to be doing in that area. The other measure of size that I had mentioned was average size. Now, you can't just take an average size of all the fish across the bay. You got to kind of look at them on a species by species basis. I looked at quite a few species, and then I pulled out just a few that were kind of representative examples that I want to present here. This is the average size of kala. It's the blue spine unicorn fish. As I understand, it's a really nice fish to catch and eat. In this figure here, it's going to offset a little bit. What we have is the circles here, which should actually be sitting down on these bars, is the average size. The bar is the standard deviation of that size. So that's kind of the variability around that. And what we have is the average size down here, so this is about, what, 12 or 13 centimeters. And then there's the sites. So these are these are a selection of the sites that TNC is actively monitoring in sort of long term. And so we have very good size data for all of these. And again, for this species, we see that Maunalua Bay sitting right down here on the bottom. When you compare it to the sites that are doing very well in terms of average size up here, you see that Maunalua Bay, the average size of fish of this species are about 1/3, maybe 1/2 of what a really sort of top site would look like. As you kind of move down, of course, it gets closer and closer. But even compared to the next closest sites here, Maunalua Bay is in almost half as large as those guys. So it's considerably smaller. We also have for uhu, this is one particular species of uhu. It's the red-lipped parrotfish. Maunalua Bay again is falling down near the bottom of this particular figure. Fish size is compared to this guy, which is what's sitting out here, about 35 centimeters on average. Maunalua Bay, at least in 2009, was sitting down around 15, so it's actually less than half the size of sort of the largest average in the state. And I want to point out that in Maunalua, in 2010, during our surveys, we actually saw no red-lipped uhu in Maunalua Bay, and that was 54. I think it was 54 survey sites. We didn't see a single one. Now that doesn't mean between 2009 and 2010 those fish disappeared. It just means that they're relatively rare out there. And in fact, in 2009, I think that's based on like 7 or 8 fish that were sighted. And I wanted to bring the uhu up because in that picture I showed you previously from the 1930s, that gentleman had a lot of uhu in his catch. Whether it was this particular species, I couldn't tell. But at one point, uhu were very common in the bay, and now seem to be a lot less common. And this is also supported by the fishermen surveys that Jack highlighted. When the fishermen noticed a huge decline in their uhu catch, and that's actually showing up in our assessments here. So, in conclusion, we're seeing that Maunalua Bay has considerably lower biomass of fish. That's both total fish and resource fish than just about any other area surveyed within the state. One exception, though, and this seems to be a little bit different than what Jack was picking up, but it looks like goatfish might actually be an exception to that. Goatfish populations seem to be comparable to a lot of other areas within the state. Some of that might be there's just a lot of sand habitat out there. Goatfish feed over the sand, so there may just be good habitat still remaining for the goatfish. In terms of fish size, the fish in Maunalua Bay on average are considerably smaller than any other areas around the state. So there's not only the decline in numbers, but there's a marked decline in size of fish within Maunalua Bay. But also the ecologically important prime spawners appear to be very rare in Maunalua Bay, at least relative to other areas. So it's kind of bad news, which is unfortunate to bring up. But following on Jack's talk, where we see fishermen and the community as a whole that seems ready and willing to make a step forward and to try and do something about the conditions here, willing to perhaps enact some management changes that may correct these conditions. Hopefully there's some bright future on the horizon that people can work towards. And hopefully this is a low point in what we're seeing in Maunalua Bay, and then actions from here we'll gradually see an upswing. TNC will be continuing to monitor in Maunalua Bay. So as some of these management actions begin to come to fruition, as things are changing on land, as perhaps sort of fishery regulations or fishery management practices are put in place, maybe we'll begin to see an uptrend in some of the fishing in Maunalua Bay. At least that's what TNC, and I'm sure all of you would hope to see. There's quite a few people who I should acknowledge. These are the guys who actually went out and collect a lot of the data. As I said, I'm not a fish person. I can barely tell you what this fish is, let alone count and size them. They do a wonderful job. In addition, there's several past TNC people who have collected fish, as well as Manuel, who currently works for TNC and works directly with a lot of you guys in the room. Alan Friedlander supplied a lot of the other sites and data is making data comparisons too. And a lot of our work is funded by these two wonderful foundations. So I'll take any questions.
Can you comment on whether it might be better to encourage the fishermen to catch 100 medium and small fish and not catch any big fish, which is just the opposite of what we do now?
Believe it or not, people have actually proposed size windows, in a sense. There might be another name for them. Slots. And to catch within those. And so effectively to leave the big ones, leave the little ones, and take those kind of medium-sized ones. I don't know enough about it myself, but based on the data I've seen, it would certainly make sense to do something along those lines. Yeah, people catch in the middle.
Well, with the data, it looks like controlled areas have more fish than areas that aren't controlled. So, are there, what's the projection of any kind of program starting in one day? Any idea?
I'm not able to really answer that question. That's something that folks in Monterey Bay have to really investigate and look at. I mean, based on Jack's sort of data, it sounds like at least the fisherman community is open to the discussion. They want to be involved in the discussion. I'm not, connected enough with the general feeling of the community, and that's something that's probably better suited to asking Malama Maunalua or just the community that's in here. But it seems like from an outsider's perspective that the Maunalua Bay general community seems like they're open and willing to at least talk about some of those things.
I don't know how fast fish grow, but if you wanted to restrict fishing so that you can give the fish a chance to get to reproductive size, how long would you have to impose a period?
Are there, we were going to ask the speakers to come up just for a few minutes and see if there's other questions. Would you mind just heading to the front and see if there's any?
Actually, this gentleman right down here, Sean, was collecting some information on coral settlements, algae settlement by putting out plates. Did you want to say anything about those?
Sure. So we'll set out some coral recruitment pits throughout sample size. I see a gentleman here smiling. I think you've seen him out there. Basically, what we try to do is provide a substrate for those organisms to settle on, since they can't settle on the mud. Provide some artificial substrate, and hopefully we should see settlement if the larvae are out there. And from our first round of collections, I haven't seen any coral recruitment or corals on any of my tiles. So we do see some crustose coralline algae, which is a good thing because corals settle on those. But then we do see a lot of other kind of fleshy algae that I think you showed a slide on spiridium. A lot of spiridium was all over my tiles. So the native species is are out there, but as far as corals, we don't see yet. Yes.
The question a moment ago is what piece of science is missing. It seems to me that one of those pieces is what the current patterns within the bay are like, because you talk about recruitment from outside Maunalua Bay, but once the basic stock is built up within the bay, one would hope that it would continue to self-recruit from that breeding stock, but we don't know whether that's even possible because we don't know anything about the current patterns. So even though the breeding stock in the bay might get up to a certain point, we might be losing all recruits to another area just because they're flushed out by current patterns.
Yeah, that's absolutely right. I do think we have some information on currents within the bay. I know Chris has some information if people want to jump online.
Can I see it on your website, what the current patterns in Maunalua Bay?
Yeah, one of the images I pulled up earlier from two means. One from actual real-time measurements and one from high-resolution model. So you can see down to your...
Can you use the microphone first, please?
Sorry. Yeah, so you can, through two different means, get at currents with real-time measurements, which we have online, and high-resolution numerical modeling.
Now, how are those current measurements made?
At the surface, we use terrestrial-based radio. So we send radio signals across the surface of the ocean, and the microscale structure of wavelets reflects that signal back. And we look at the Doppler shift in the radio frequency, and we can infer how fast the currents are moving towards or away from the sensors. And we have two of them that overlap, so you can actually get two-dimensional current velocities.
Within the,
Not all the way in, definitely within the sort of demarcation line between Port Rock and Diamond Head, but not into the surf zone. We don't have the technology to be that highly resolved.
USGS was out here, maybe 2 years ago now, and they deployed a whole bunch of sensory equipment right in Maunalua Bay, and they did some current modeling within the bay also. I don't remember exactly what their results were, but they were looking at currents in relation to sediment movement, and what they found was, if I remember correctly, it's kind of a sloshing of the current back and forth. I don't want to say too much more because I might say something wrong at that point. But that information is actually available in an open file report, which I have to give you my card, so you can email me and then I'll dig that information out if it exists.
I can, the hat I normally wear is an oceanographer and a physical oceanographer, and I can say that along the South Shore, Dwayne's exactly right, currents tend to oscillate just back and forth with tides. The tidal currents are the dominant driver of circulation in the near shore. So generally, sediment and larvae and water just move back and forth with very little long-term progress along the shoreline.
so then we would expect that if there is a sufficient brood stock in Maunalua Bay, it'll self-recruit in those currents.
Certainly it's a possibility, or those recruits may just slowly transfer down the shoreline, and you may get recruitment from Anama Bay or from Port Lock moving down that coastline.
Just one more thing to add to Jared's question about what science is missing. Well, what I didn't see here is when the breeding or the spawning patterns are for fish, and I've seen graphs out there before, but they haven't been based in Maunalua fish. So if we knew when each species was breeding, from my understanding of the ancient Kapu system was that they wouldn't get fish when they're breeding because that would be counterproductive. So one piece of science I'd like to see is what the spawning calendars are for target fish in Maunalua.
I'd like to thank our presenters for the huge effort that they have made and the information they've brought to this community. We are really, truly grateful. Can we have a round of applause?